So hi guys and welcome back to Excelsior Academy. So new topic over here is cell walls, the extracellular matrix and the cell interactions. The cell wall is something which we'll be covering today, and then later we'll cover the extracellular matrix as well as the cell interactions. Now when we talk about the cell walls, so although the cell boundaries are defined by the plasma membrane, so many cells are also surrounded by insoluble array of the secreted molecules, right? So in the case of bacteria, fungi, algae, higher plants, we know that there is an additional, that is the cell wall is present. So everyone knows like the cell wall of bacteria is made up of what? Of fungi, of algae, of plants, right? And we also know that the animals does not have the cell wall. Okay. So although animal cells are not surrounded by cell walls, most of the cells in animal tissues are embedded in the extracellular matrix consisting of secreted proteins and the polysaccharides. So we'll come to this. This is really very important and interesting also to study. So it does not only provide structural support to the cells and tissue, but also play important role in regulating the cell behavior. Since just now we saw that the integrins, it is connected to it, right? So any changes which happens in the extracellular matrix, it is just like a signal to the cell, and accordingly, based on the signal received by the cell, it is going to behave. So interactions of cells with the extracellular matrix anchor the cytoskeleton and regulate the cell shape and movement. So the extracellular matrix gives, and suppose let's say that it gives a signal ki bhago yahan se. So what will happen? So cytoskeleton proteins, both actin actin filaments and microtubules, they will ensure that the cell starts moving in the ahead, like in the forward direction, right? Likewise, direct interactions between the cells are key to the organization of cells and tissues, as well as providing channels through which cells can communicate with their neighbors too. Now, first topic is about the cell wall. So today we'll complete the cell wall topic. So the rigid cell walls that surround bacteria and many types of eukaryotic cells determine the cell shape, prevent the cells from swelling and bursting as a result of osmotic pressure. Okay, despite the common function, the cell walls of bacteria and eukaryotes are different. So we already know about it. Now coming to the bacterial cell wall. So rigid cell walls of bacteria provide protection against osmotic pressure and determine the shapes of the different kinds of bacterial cells. So now we will know why these spherical bacteria are spherical in shape, why the rod shaped bacteria are rod in shape. So the reason the shape which is determined, okay, will come to know in this. For example, we know that E. coli is rod shape. Pneumococcus, Staphylococcus are spherical in shape, whereas spirochete, they are spiral in shape. So why they are spiral, why they are spherical, or why they are rod shape will come to know in this lecture. So because of this cross-linked structure, the peptidoglycan forms a strong covalent shell around the entire bacterial cell. So now we are going to discuss about the peptidoglycan. So I'm sure that everyone knows what is peptidoglycan layer and how it is different in the gram-negative and gram-positive bacteria. Okay, before I proceed, who can tell me that whether that the peptidoglycan is thicker in gram-positive or gram-negative? Yes, I repeat my question. The peptidoglycan layer is thicker in gram-positive or gram-negative bacteria?
It is thicker and positive.
Very good. It is thicker in positive. Good. And this peptidoglycan is made up of alternating units of NAM and NAG. So you know what is NAM and NAG. They are N-acetyl glucosamine and N-acetyl muramic acid, right? And the linkage between NAM and NAG is beta-1,4-glycosidic linkage. Though you don't have to remember the structure, but you have to remember like what kind of linkage is present between NAM and NAG. So this is beta-1,4 linkage. Okay. Now we also know that these NAM-NAG, okay, this chain are also cross-linked with the help of the tetrapeptides. So even that tetrapeptide you have to remember. So that is the tetrapeptide where you see the existence of D amino acids. Though we know that only L amino acids are present, but in nature, but bacteria, it has got D amino acids also. Now coming to the difference between the gram positive and gram negative. So let's go with the gram negative. So we see that in the case of gram negative, the peptidoglycan layer, okay, it's less. This is the peptidoglycan layer. Now, how the gram-negative bacteria is getting the extra protection is because of the outer membrane, which has got LPS, that is lipopolysaccharide. Okay, so because of this, it has got the extra protection, though it has the thinner level of or layer of peptidoglycan layer, and this LPS will have all the porins of bronze lipoprotein, which is going to cause the infection wherever it is getting accumulated. Now, coming to the gram-positive bacteria. So we know that in this case, the peptidoglycan is very thick, and it also has got the lipoticoic acid and the teichoic acids. Okay, the presence of this. So I'm sure this you all guys know the difference between the the difference between the cell structure of the gram-negative as well as the gram-positive bacteria. Okay, I'm not going in detail. Okay, so this we also know that the peptidoglycan layer is made up of NAM and NAG, alternating units of NAM and NAG, and then we have got the peptide chain, which is getting in between the in between the NAM and NAG chain. Okay, so now coming to the synthesis of peptidoglycan, this is something important. Okay, I am not asking you to remember this but you need to understand what steps are important. Okay, so again, NAM and NAG, fructose 6-phosphate. So this is from where you're getting from the glycolysis, right? So all those steps in detail we are not going. Just understand that from glycolysis byproduct fructose 6-phosphate, it is getting synthesized. Okay, so glucosamine 1-phosphate acetyl CoA is being added, and then you get it unacetyl glucosamine 1-phosphate. Later you get UDP NAG. Okay, so ये UDP क्या है? You have got those carriers. UDP choline, all those carriers are there, which comes. Okay, now UDP NAG, then again all others. From UDP NAG you get your NAM. Okay, and then later all those amino acids are added one at a time to form the pentapeptide. Now this is the peptidoglycan cell wall synthesis. Okay, thoda sa tricky hai, but till now nobody has asked a question based on the peptidoglycan synthesis, but it is always better to know like how the antibiotics are attacking. Okay, how they are playing important role. So if possible, try to remember or try to understand like cycloserine हो गया, vancomycin हो गया, या bacitracin हो गया. So these are known, very common known antibiotics, correct? So these where and all it is getting acted upon, that is what you have to remember. Okay, now let's go slowly about the peptidoglycan cell wall synthesis. Okay, the first thing is that UDP derivatives of NAM and NAG are synthesized. So here it is not shown. That's why in the earlier slides I mentioned like how the NAM and NAG are synthesized. So once you have this, then what? UDP NAM is there. Okay, now what is happening? Sequential addition of amino acids to form the NAM pentapeptide. ATP is used. Okay, now but tRNA ribosomes are not involved in forming the peptide bonds that link the amino acids together. What does it mean? So these amino acids are already present over there. Okay, and हम यहाँ पे कोई भी ribosome tRNAs नहीं use कर रहे हैं to form the peptide bond formation. Okay, so here it is the based on the energy required or taken up by the enzymes to synthesize. Okay, so UDP NAM हो गया. Then you have got this peptide. तो पहले आपका alanine आया, glutamic acid आया, lysine आया, and then 2 molecules of D-alanine. Okay, now you can actually D-alanine कहाँ से आ रहा है? Obviously आपके L-alanine से आ रहा है, which is getting isomerized to D-alanine. These steps can be inhibited by your cycloserine. Is this part is clear, like how UDP NAM pentapeptide is formed? Yes. So I'll go in one stretch. Please stop me in between, okay? If any of the step you're not able to follow. Okay. So I hope this much is clear, like from UDP NAM. Okay. Now why NAM NAG is not coming directly? Because if suppose they are kept free, so they are very reactive. They might form covalent bonds with the other molecules present. So that's why UDP as a base, it is present in order to provide NAM-NAG molecules towards the cell wall synthesis. Okay. Okay, so NAM is a pehla alanine, glutamic acid, lysine, and 2 molecules of D-alanine. Okay, so ये 3 और ये 2 5 हो गए आपके. So UDP NAM pentapeptide is formed. Okay, now this entire thing is being transferred to your bactoprenol. Okay, now what is bactoprenol? Looking at the structure, sorry, looking at the position, can you tell what is bactoprenol? It's nothing but the phospholipid type, okay, which is present specifically for your bacteria. Okay, so now it is being transferred to your bactoprenol. UDP NAM transferred to your bactoprenol. Now bactoprenol double phosphate NAM pentapeptide. So what do we see here? This UDP, okay. So bactoprenol was already phosphorylated. Another phosphate group has been added from the UDP. So UDP becomes now, uridine diphosphate becomes now, one phosphate is being added, right? Here, another phosphate is being added. So UDP becomes your UMP. And this entire thing, NAM pentapeptide along with the phosphate is added or transferred to the bactoprenol. So bactoprenol, phosphate, phosphate, NAM pentapeptide. So this is your first lipid which is formed. Okay, the NAM molecule is ready. Now comes the UDP NAG. Okay, now UDP NAG is there, sorry. So what is happening? This will come, this will join over here. So NAM NAG has been added. यहाँ पे आपका NAG add हो गया है. And then you have got the pentapeptide also. Is it clear, guys? This much till here? Everyone? No, not able to follow.
Ma'am, can you just repeat once?
Okay. See, एक बार यहाँ तक समझ में आ जाएगा ना, then it becomes easy. Okay, so I'll go once again. Okay, so first in the first few slides we saw that how UDP NAG से आपका UDP NAM बना, ठीक है? Now, but for the peptidoglycan cell wall synthesis, we need NAM first. तो सबसे पहले क्या हुआ? आपका UDP NAM आया. To this first your pentapeptide will be added. What are these pentapeptide which is involved in the cross-linking? ठीक है? We'll come to cross-linking later. So the sequence is also important. First your alanine, then glutamine, then lysine, and then 2 molecules of D-alanine. Okay. Now D-alanine is coming from your L-alanine. Okay. Now these are being synthesized, these are being joined. Of course, it's a peptide. So UDP NAM pentapeptide has been formed. Now this UDP NAM pentapeptide is being transferred to the bactoprenol, which is your bacterial lipid. Okay, now this has been transferred to the bactoprenol. So bactoprenol, when it was taken, okay, it was having already the phosphate group. Now UDP becomes UMP by transferring phosphate group to the bactoprenol. So bactoprenol becomes double phosphate plus NAM and pentapeptide. Then NAG unit comes, it gets attached to this. Okay, so this is your 1 unit of peptidoglycan. Now this is being transferred to the outer layer, okay, outer layer of your cell wall. So what is happening then? The NAM-NAG pentapeptide is then attached to the growing peptidoglycan layer, which is again increasing the length of your peptidoglycan. Okay, are you getting it?
Yes, yes.
हाँ, and later then what will happen? Bactoprenol का काम हो गया तो अभी वो वापस जाएगा, फिर से दूसरा यूनिट ऑफ NAM-NAG लेकर आएगा, ठीक है? And then later this peptide pentapeptide जो है, it will get cross-linked by the transpeptidation action, which will be shown in the next. Just go through, I'll just remove these highlights. Just go through yourself all the steps and let me know if any steps are not clear. Okay. We are good. And then we saw that there are two antibiotics, cycloserine, vancomycin. Vancomycin is inhibiting the step of addition of peptidoglycan units. And then we have got the bacitracin. Bacitracin is inhibiting the transfer of bactoprenol for the further cell wall synthesis. Okay. Cool. Shall we move ahead?
Yes.
Okay. Then comes the transpeptidation, that is cross-linking. So if it is a gram-negative bacteria, then it is your between the tetrapeptide, the difference. Okay. Ala, glutamine, then lysine and D-alanine is there, right? Oh, sorry. This cross-linking is between the, in the E. coli, it is between the, here they form only the, the typical cross-linking which is formed, okay, over here. It's again the peptide. ये हो गया आपका पेप्टाइड, ये भी आपका क्रॉस लिंकिंग जो है वो भी आपका पेप्टाइड ही है, ठीक है. नाउ कमिंग टू द ग्राम पॉजिटिव, हियर अगेन सी only glycine they have used in order to make the cross linking between the two chains of the peptidoglycan. Okay, so in the case of gram negative and gram positive, this inter cross linking is different. Now interestingly, the unique structure of their cell walls also makes bacteria vulnerable to some antibiotics. Already we saw certain antibiotics, right? So that was the example. Those antibiotics are secreted by the other bacteria. Okay, so whenever two different species of bacteria are present, in order to survive, okay, for themselves to survive, to get all the resources, they start secreting out the antibiotics. So sometimes these antibiotics are lethal to the other species of the bacteria. For example, just now we saw vancomycin and bacitracin are there, right? Now we have penicillin. So you know penicillin kahan se aapka derive hua hai. So it also have the ability to inhibit the enzyme responsible for forming the cross-linking, यानी कि ट्रांसपेप्टिडेशन जो होता है उसको वो इनहिबिट करता है, and thereby interfering the cell wall synthesis and blocking the bacterial growth. Now let's see how this is happening. So you have got this peptidoglycan, correct? So just now we saw that NAM, NAG are there, and these pentapeptides are present, correct? So the cross-linking which happens between this pentapeptide with the help of the transpeptidase enzyme, okay, that is being blocked. And penicillin comes in the category of your beta lactam. Now, why I'm going with this is that because when people found out like how penicillin, which is being secreted by a fungus, is able to kill a bacteria by inhibiting a cell wall synthesis. कैसे? तब पता चला that the structure of beta lactam ring, which is present in the penicillin, is actually sitting very nicely in the active site of your enzyme called transpeptidase. Okay, so I think, yes, I have a video over here to understand how exactly this is happening. Let's watch this. Okay.
Structurally, most bacteria consist of a cell membrane surrounded by a cell wall, and for some bacteria, an additional outer layer. Internal to the cell membrane is the cytoplasm, which contains ribosomes, a nuclear region, and in some cases granules and/or vesicles. Depending on the bacterial species, a number of different external structures may be found, such as a capsule, flagella, and pili. In gram-negative bacteria, the gap between the cell membrane and the cell wall is known as the periplasmic space. Most gram-positive bacteria do not possess a periplasmic space, but have only periplasm, where metabolic digestion occurs and new cell peptidoglycan is attached. Peptidoglycan, the most important component of the cell wall, is a polymer made of N-acetylmuramic acid, alternating with N-acetylglucosamine, which are cross-linked by chains of 4 amino acids. The function of the bacterial cell wall is to maintain the characteristic shape of the organism and to prevent the bacterium from bursting when fluid flows into the organism by osmosis. Synthesis of the peptidoglycan and ultimately the bacterial cell wall occurs in a number of stages. One of the first stages is the addition of 5 amino acids to N-acetylmuramic acid. Next, N-acetylglucosamine is added to the N-acetylmuramic acid to form a precursor of peptidoglycan. This peptidoglycan precursor is then transported across the cell membrane to a cell wall acceptor in the periplasm. Once in the periplasm, the peptidoglycan precursors bind to cell wall acceptors and undergo extensive cross-linking. 2 major enzymes are involved in cross-linking: transpeptidase and D-alanine carboxypeptidase. These enzymes are also known as penicillin-binding proteins because of their ability to bind to penicillins and cephalosporins. Eventually, several layers of peptidoglycan are formed, all of which are cross-linked to create the cell wall. Gram-positive bacteria may have more layers than gram-negative bacteria and thus have a much thicker cell wall. Beta-lactam antibiotics include all penicillins and cephalosporins that contain a chemical structure called a beta-lactam ring. This structure is capable of binding to the enzymes that cross-link peptidoglycans. Beta-lactams interfere with cross-linking by binding to transpeptidase and D-alanine carboxypeptidase enzymes, thus preventing bacterial cell wall synthesis. By inhibiting cell wall synthesis, the bacterial cell is damaged. Gram-positive bacteria have a high internal osmotic pressure. Without a normal rigid cell wall, these cells burst when subjected to the low osmotic pressure of their surrounding environment. As well, the antibiotic penicillin-binding protein complex stimulates the release of autolysins that are capable of digesting the existing cell wall. Beta-lactam antibiotics are therefore considered bactericidal agents. Bacterial resistance to beta-lactam antibiotics may be acquired by several routes. One of the most important mechanisms is through a process known as transformation. During transformation, chromosomal genes are transferred from one bacterium to another. When a bacterium containing a resistance gene dies, naked DNA is released into the surrounding environment. If a bacterium of sufficient similarity to the dead one is in the vicinity, it will be able to uptake the naked DNA containing the resistance gene. Once inside the bacterium, the resistance gene may be transferred from the naked DNA to the chromosome of the host bacteria by a process known as homologous transformation. Over time, the bacterium may acquire enough of these resistance genes to result in a remodeling of the segment of the host DNA. If this remodeled DNA segment codes for cross-linking enzymes, i.e. penicillin-binding proteins, the result is the production of altered penicillin-binding proteins. These altered penicillin-binding proteins can still cross-link the peptidoglycan layers of the cell wall, but have a reduced affinity for beta-lactam antibiotics, thus rendering the bacterium resistant to the effects of penicillin and other beta-lactam agents. This transfer process has resulted in penicillin-resistant S. pneumoniae through the acquisition of genes from other naturally occurring penicillin-resistant Streptococcus species. A second important mechanism by which bacteria become resistant to beta-lactam antibiotics is by the production of enzymes capable of inactivating or modifying the drug before it has a chance to exert its effect on the bacteria. Depending on the bacterial species, the gene coding for these enzymes may be found as part of the host DNA or on plasmids, which are small, self-replicating units of genetic material. Bacteria are capable of passing these resistance plasmids to each other by conjugation. When 2 bacteria come into close contact with each other, a small channel is created between them which allows one of the bacteria to pass a copy of the resistance plasmid to the other. If the plasmid is transcribed and translated, the bacteria will begin to produce inactivating enzymes. These enzymes, capable of destroying beta-lactam antibiotics, are known as beta-lactamases. In gram-positive bacteria, the beta-lactamase enzyme is generally inducible, resulting in a large amount of enzyme being produced in the presence of the drug. In gram-negative bacteria, the beta-lactam enzymes are produced constitutively, i.e., even when the antibiotic is not present. Gram-positive bacteria release the beta-lactamase enzyme from the cell into the extracellular environment where it inactivates the drug before it enters the bacterial cell. In contrast, gram-negative bacteria retain the beta-lactamase enzyme within the periplasmic space, resulting in a more efficient mechanism than gram-positive bacteria. Ultimately, the destruction of the beta-lactam ring of the antibiotic renders it incapable of binding to the penicillin-binding protein, and thus the bacteria become resistant to that drug or class of drugs.
Okay. Did you get it? थोड़ा सा extra भी था. Hello? Did you get that? Like how the penicillin beta lactam is actually, inhibiting the cell wall synthesis, and of course it was little extra also, like how the bacteria are able to get the resistance, right? So there were two types. Either you modify the transpeptidase or you secrete beta lactamase. Okay. Now coming to the cell wall growth. The cell wall growth is continuously enlarged during the bacterial growth and division. So the maintenance of the characteristic shape of the bacterial cell is also important. Okay. So this is determined by the bacterial cytoskeleton protein. So this is what I was telling in the beginning, like why the spherical shape bacteria are spherical in shape, why rod shape. So all these proteins, okay, which are responsible for determining the shape of the bacteria are the cytoskeletal proteins. So the first cytoskeletal protein is FTSZ, which is homologue of eukaryotic tubulin, okay, which is found in nearly all bacteria, which is present in spherical, rod shape and spiral shape. So FTZ is present in all, okay. Now it forms a ring-like structure at the site where the cell division occurs, directing synthesis of the new cell wall that separates the newly formed daughter cells. So जहां पे आपका cell divide हो रहा है, there your FTSZ is present. Now in the case of the rod-shaped bacteria, you have got another protein, cytoskeletal protein called MRB. Okay, so this is the homologue of actin. Now, this is directing the cell wall synthesis during the cell elongation. So, as you see, in the case of the spherical shaped bacteria, it is the spherical shaped bacteria, it is the presence of only FTZ. In the case of the rod shape, it is FTZ and the MRB. Now, if it is a spiral shape, then you have got another protein called crescentin. Okay, so all these are responsible for bringing the shape in the respective bacteria. Get it, all of you? Shruti, is it clear?
Yes, ma'am.
Okay. Now let's come back to our MCQs. Okay. Are you ready, guys? You have to be ready. I told you yesterday, right? So let's continue. Very few questions are there. Let's complete in this remaining time. So the first question is, cystic fibrosis transmembrane conductance regulator is known to control the transport of which ion?
Chloride.
Very good, chloride ion. Next question. The beating of cilia is regulated by, this is not action, okay? This is actin. Actin, myosin, cofilin, nexin. So what is the answer?
Actin.
Actin, very good. Yes. The next question. बहुत छोटे-छोटे से simple से हैं. Now, in an attempt to study the transport of secretory vesicles containing insulin along microtubules in cultured pancreatic cells, how would treatment with colismid affect the transport of these vesicles? So remember the effect of the colismid. Colismid क्या करता है microtubules के साथ? Okay, so options are Coulsomid induces polymerization of microtubules, which in turn would activate vesicular transport. Polymerization of microtubule is inhibited by coulsomid, which in turn would inhibit the transport of secretory vesicles. Coulsomid inhibits the vesicular trafficking through inactivation of V-snare proteins. Coulsomid activates T-snare protein and in turn activates vesicular transport. Yes, what is the answer, guys? Akshaya, क्या आंसर होगा? Even if you don't know the what to say, the action of coulsomid, then also you should be able to answer this. Okay, Krishita, would you like to try?
I guess B, polymerization of microtubules is inhibited by coulsomid, which in turns would inhibit the transport of.
Correct, correct. That's the answer. See, coulsomid is a drug, okay, which we supposed to stop or prevent microtubule formation. Coulsomid ho gaya, colchicine ho gaya, right? That's how you were able to visualize the mitosis stages of your cell, okay, in the lab. So coulsomid is also, अब ये क्या बोल रहा है? Coulsomid is inducing polymerization and helping in the activation of the vesicular transport. नहीं, ऐसा कुछ नहीं है. और ये V snare T snare का तो कोई रोल ही नहीं है coulsomid के साथ, ठीक है? So the answer is B. Polymerization of microtubule is inhibited, hence inhibiting the transport of the secretory vesicles. Good. So the correct answer is B.